Hello all, welcome everyone. I'm Kim from Tuskegee University and I'm going to present the research article by Gugliandolo et al. 2021 on the topic of effect of cannabidiol CBD on canine inflammatory response and ex vivo study on LPS lipopolysaccharides stimulated whole blood. And this is for the graduate seminar second, and our instructor is Dr. Jackson. Introduction. CBD is one of the major non-psychoactive plant-derived cannabinoids, and it can be found in most of the cannabis species, and it is known to act as an anti-inflammatory and immune-modulant agents, and this CBD can affect both innate and adaptive immune response. The rationale of the study is that immune modulating, anti-inflammatory and other biological activities of the CBD has been characterized most often in the human medicine, but there are very few studies in the veterinary medicines, and the pharmacokinetics and tolerability of the CBD has already been characterized in the dogs, but there are very few studies on the therapeutic use of the CBD in dogs. So the objective of this study was to evaluate the effect of CBD directly in the canine inflammatory response. So to get this objective, the ex vivo whole blood culture was done with the stimulation by lipopolysaccharides, focusing on the level of major regulating cytokines, pro-inflammatory cytokines, that is IL-6 and TNF-alpha, and anti-inflammatory cytokines IL-10. In this study, the level of NF-kB, the nuclear factor Kb, which regulates the inflammatory response and transcription of the other pro-inflammatory cytokines like IL-6 and TNF-alpha, and the COX-2 expression, which plays the fundamental role in inflammatory cascades, were also studied. For this study, the whole blood from 6 healthy dogs were collected, and these 6 dogs were German Shepherd breed, and they were from 2 to 10 years old, and 3 were male and 3 were female. So the whole blood culture and stimulation was done for 24 hours. For this, there were 4 groups. The first group is control group. In control group, the whole blood was cultured only with the vehicle. The vehicle was DMSO 0.1%. In second group, it was LPS group, and in this group, whole blood was stimulated with LPS 100 nanogram per ml and cultured, and the vehicle use was DMSO 0.1%. The next group was CBD 50 microgram. In this group, whole blood was cultured by stimulation with LPS of 100 nanogram per ml, and it was treated with CBD 50 microgram per ml. The fourth group, the last group, is CBD 100 microgram, and in this group, whole blood was cultured and stimulated with LPS 24 nanogram per ml plus CBD 100 microgram per ml. In this study, the level of IL-6, TNF-alpha and IL-10 in the whole blood culture supernatant was measured with the help of sandwich ELISA kit from the Quantkine R&D system company, and the RT-PCR technique was used to measure the mRNA level for COX-2. Additionally, the Western blot technique was used to measure the expression level of NF-kB, P65 and COX-2 protein. And here is the results and discussions. The first graph shows the level of IL-6 in the different treatment groups. In the group with no LPS treatment and no CBD treatment, the IL-6 level is quite lower, and in the second group with LPS stimulation without CBD, the IL-6 level increased significantly, and in the third group with LPS stimulation and with the CBD treatment of 50 mg per ml, the IL-6 level decreased significantly, and similarly in the fourth group with LPS stimulation and CBD treatment of 100 mg per ml, the level of IL-6 again decreased significantly. And the next graph shows the TNF alpha level in the different groups. Here we can see as the CBD treatment increases, the TNF level significantly decreased. And as we know that the IL-6 and TNF-alpha plays the key role in physiological inflammatory response, such as during the infection, and are also associated with the poor clinical outcome. And here results showed that the significant lower level of secretion of IL-6 and TNF-alpha when treated with the CBD. Here in the first graph we can see the level of IL-10 in the different treatment groups. In the first group, without LPS stimulation and without CBD treatment, the IL-10 level is little lower, and in the second group with LPS treatment and without CBD treatment, the level of IL-10 increased significantly. But in the third group, the level of IL-10 is increased, but it's not significantly different than this second group. And in the fourth group also, the level of IL-10 increased, but it is not significantly different than the second group. In the second graph, we can see the different ratio of TNF-alpha to IL-10 in the different groups. In the first group, the ratio is little bit lower, but in the second group with LPS treatment and without CBD, the TNF-alpha to IL-10 ratio is highly increased. But in the third group with LPS treatment and CBD treatment, the level of the ratio decreased significantly, and in the fourth group also the ratio decreased significantly. The level of IL-10, which is an anti-inflammatory and immune modulating cytokines, was not significantly altered by the CBD treatment. And as in the maintaining the immune homeostasis, the level or the balance between TNF-alpha to IL-10 ratio is very important, because an increase in TNF-alpha is counterbalanced by the simultaneous increase in the anti-inflammatory cytokines IL-10. So the ratio between TNF-alpha and IL-10 ratio is very important, as the results show that LPS stimulation can produce significant increase in TNF-alpha to IL-10 ratio, while the treatment with CBD is able to significantly reduce the ratio. Thus, this is desirable effect. And here the first picture is the result of Western blot analysis for the NF-κB expression. We can see the increase in NF-κB expression in the second group with LPS stimulation but without CBD treatment, and in the third and fourth group with CBD treatment, the level of NF-κB P65 decreased. And in the second graph also shows the similar thing. You can see here the level of NF-κB increased significantly with the LPS stimulation and without the CBD treatment, but with the CBD treatment the level of NF-κB goes on decreasing significantly. The NF-κB induces the expression of several pro-inflammatory genes, and results show that the NF-κB P65 expression was reduced when treated with CBD. And this finding is consistent with the decreased level of pro-inflammatory cytokines IL-6 and TNF-alpha in this experiment. In the first picture, you can see the expression of COX-2, and here we can see the increased expression of COX-2 with the LPS stimulation but without CBD treatment, and with the CBD treatment the COX-2 expression goes on decreasing. In the second picture we can see the similar result. We can see here the level of COX-2 increased significantly than the control group when LPS stimulated and without the CBD treatment, but with the CBD treatment the level of COX-2 goes on decreasing. Similarly, the mRNA fold change is shown in the third picture. You can also see the mRNA of COX-2 increased significantly with the LPS stimulation, but it decreased with the CBD treatment. And COX-2 is the key enzyme in synthesis of the prostaglandins from the arachidonic acid, and it plays a key role in inflammatory response. Both COX-2 mRNA and COX-2 protein are significantly increased by the LPS stimulation in this research, but they are significantly reduced when the blood culture are treated with the CBD. So despite of having the very significant findings in this research, there are some of the limitations. In this study, there are no accurate study on the cytotoxic effect of CBD if present, and thus no indications whether the doses used in this study could fall within a therapeutic range or not. Similarly, there is a limited use of CBD concentration, and there is limited sample size, and there might be the species difference in the response with the LPS and CBD treatment. And the next limitation is that the source and preparations of the CBD may vary, so its effect may also vary, which is not studied in this research. So as a conclusion, the CBD was able to reduce the inflammatory response induced by the LPS in canine whole blood. Thank you.